We will wrap up our discussion of the genetic processes of evolution with some more talk about interactions among genes, natural selection, adaptive landscapes, and the evolution of genomes in the largest sense. Classic Mendelian traits are ones that exhibit discontinuous variation. That is, that there is a clear difference between dominant and recessive phenotype when there is a minimum of environmental influence. Most traits of interest for us and that are relevant to fitness are actually polygenic in nature, though, and those polygenic traits appear as a continuous range of variation rather than as discrete categories. Strictly speaking, for polygenic traits, there are 2n plus 1 phenotypes. N indicates the number of genes. In this case, for 3 genes related to the production of melanin, a person can have from 0 to 6 doses of a dominant allele, resulting in 7 categories of skin coloration. On the other hand, remember that there is also a strong environmental contribution to this phenotype as well. When we look at 5 genes that interact to create a common trait, we jump all the way to 51 different phenotypes, and that is ignoring any environmental contribution at all, so this probably also would appear as continuous variation. In practice, not all allelic combinations occur with equal frequency. Natural selection can have the effect of revealing the latent variation for a character. For example, in this population of Galapagos finches, we can compare the average depth of the parent's beak with the average depth of the offspring's beak. The positive slope on this regression shows that overall, parents with deeper beaks also have offspring with deeper beaks. This might seem like kind of a random trait, but beak depth is related to feeding behavior and the size of the food items that can be cracked by the bird. Looking at the data on the right, first we see the overall bar chart that represents the distribution of beak depth within the population, from a low of 7.3 to a high of 10.8. The darker portions of the bars indicate which birds actually had offspring. This graph demonstrates that the larger beak sizes tended to reproduce more often. The lower graph shows the distribution of all the offspring from that year. In this case, the average value for beak depth overall went from 8.8 to about 9.5 millimeters, so we can identify this microevolutionary change for the trait in the population. However, notice that in that 1978 cohort, more individuals have larger beak depths, and some individuals had even deeper ones than were present in the 1976 population. This increased range of phenotypes reveal the latent variation that is caused by the shifting of allele frequencies by genetic reassortment. These new phenotypes come from the pre-existing variation rather than by mutation. We know from our genetics classes that genes that are close to one another on the same chromosome tend to travel together during the process of meiosis and gamete production. Because they are physically close, it's unlikely, or at least less likely, that crossing over will recombine them into different haplotypes. Haplotypes refer to sets of genes that tend to be inherited together because of their physical linkage. They are the complement of alleles that are found in a gamete, and they are differentiated from one another by the occurrence of single nucleotide polymorphisms in the DNA sequence. The occurrence of these haplotypes in proportions different than what would be expected by Hardy-Weinberg equilibrium results in a condition that we would call linkage disequilibrium. Linkage disequilibrium is the measure of evolution in allele frequencies that comes about through evolution. We won't get into many details with that here, but understand that linkage disequilibrium happens most in cases when we have small populations that are undergoing evolutionary change, whether through drift or selection, non-random mating, or migration. These, again, are the assumptions that we invoke in the Hardy-Weinberg null model. As before, the genetic differences that accumulate through evolution are undone through gene flow, undone by bringing genes from other populations to be combined to a larger gene pool. Interestingly, haplotypes evolve not only through selection, but also through a phenomenon called genetic hitchhiking. In this case, unrelated traits that are either neutral or harmful to fitness are brought along with other traits that are selected for as another consequence of physical linkage. Hitchhiking also helps us to identify recent positive selection. It also causes a condition called genetic draft, in which an unrelated trait rides along with traits under positive selection. For example, here I show the association between temperament, patchiness of fur coloration, and ear posture among a cohort of domesticated animals that has also been shown experimentally in Russian silver foxes. Population geneticists can study changes in allele frequencies quantitatively, but it is often common for evolutionary biologists to describe more metaphorical descriptions of evolution through Sewell Wright's adaptive landscape or fitness landscape model. The adaptive landscape is a map that compares phenotypes with fitness. The XY position on the map is determined by the combination of traits. When thought of as a volume, the elevation of the landscape represents the relative fitness of each part of the volume of the metaphorical fitness space that is created. Populations would occur as point clouds in this 3D fitness space. As natural selection chooses successful combinations of phenotypes, we would expect to see populations climb those adaptive peaks over generational time. In a static landscape, as on the left, one in which the force of selection for that combination of phenotypes is stable, populations would tend to keep that population at those peaks unless the rules of selection changed, because stabilizing selection would tend to maintain the phenotypic average. On the other hand, when the environment does change, as was the case of the Galapagos finches and their beaks, populations would travel up the new peaks. If the fitness maxima would oscillate between two peaks, the populations would mirror those changes and the individuals better fit for the next peak would travel up it. Extinction happens in populations when they can't get up those peaks fast enough because of the limits of the phenotype or generational time or other factors. In a static fitness landscape, populations would tend to stay on their local fitness maximum, but it's unlikely that they would jump from one to another without significant mutation. And that level of mutation is usually harmful rather than beneficial. It's also not required that every population be at the global fitness maximum. Consider the example of the vertebrate eye again. One contingency of the history of life is that the lineage that led to vertebrates ended up evolving an eye in which the retina is wired backward compared to what you would expect for an eye. Yes, we can see, but light energy has to pass through 3 layers of nerve cells before it gets to the layer where it's absorbed, so brighter light is necessary in order to stimulate the photoreceptors. Another consequence is that vertebrates have a blind spot on the retina in the place where the optic nerve is formed by the convergence of nerve cells. Cephalopods and their relatives also have a cameral eye with a lens, but their retinas are wired what we would consider to be the right way around. They send multiple optic nerves that diverge after the retina to send information, so their eyes are inherently more sensitive to low light than ours are, and they don't have blind spots. But could vertebrates evolve such an eye? Probably not. Richard Dawkins uses the metaphor of the auxiliary powered outlet in cars. Sure, today our cars have USB ports and stuff, but back in the day we only had that round 12-volt DC outlet. Your parents or people of my generation would probably still refer to it as the cigarette lighter. There's nothing inherently good or bad about that round outlet, but it's came into place because some mechanic somewhere made a decision to put it in a car and it ended up becoming universal. We've altered it a little so that you can plug other cords to charge your phone or other gadgets, but that's an example of backward compatibility of an existing standard. In evolution, we would call that an exaptation, something that evolved first for one function, but was pre-positioned to take on a new one. Sometimes people talk about exaptations as being pre-adapted, but I don't love that terminology only because it sounds teleological or forward planning. But it's okay as long as you remember that there's no intentional design in evolution, only selection from existing variation. So, a similar case could be made for the evolution of our crappy backwards retinas as for the cigarette lighter. There was a mutational change that happened, and our ancestors just worked with it to try to optimize it for vision. Couldn't we just re-evolve a better eye? The adaptive landscape model helps us to visualize why this is unlikely. During the evolution of animal eyes, there was this contingent mutation that took place in the eyes of the ancestor of vertebrates. but our retinas work pretty well. So in order for selection to evolve improvements to the way that it works by independently evolving an eye like cephalopods, it's likely that we would have to go through a number of stages in which vision is actually less good than it is now because of how mutations tend to be deleterious. So vertebrates are basically stuck on this local optimum, but cephalopods might be occupying a global optimum. There are some groups of vertebrates, like birds of prey, that have evolved additional eye structures that allow them to maximize their visual acuity, but it's done within the constraints of this existing retinal plan. The pecten is a circulatory structure that hyperoxygenates the retina in order to decrease those nervous cells' latent period. Let's switch gears to talk a little about patterns in evolutionary genomics. Even before the time of rapid whole genome sequencing of species in the early part of the 21st century, evolutionary biologists recognized two paradoxes in regard to the evolution of genomes. The first is the C-value paradox. The C stands for copy number. We would predict that organisms with large genomes would tend to be phenotypically complex, but there are many exceptions, particularly among green plants. My favorite example is the whisk fern Psilotum nudum, which for all the world looks like a green stick, but has a genome of 2,500 billion base pairs. What does this fern do with 100 times more DNA than us? Not much. What the whisk fern has is many, many whole copies of its entire genome. That's what gives it such a large genome, just raw duplication of DNA. This phenomenon was partly understood by the discovery of non-coding DNA. DNA that doesn't code for functional polypeptides comes about by various mechanisms, including mutation into pseudogenes by our own genes, and also by the insertion of retroviral DNA into our genome. DNA inserted by retroviruses become as much of our genome as those inherited from our ancestors. If they are present in germline cells that make the gametes, they'll be passed down as such. Transposons, like sign elements, are sources of non-coding DNA as well. These segments of DNA get themselves replicated and inserted far and wide in the genome. Both of these examples, as well as introns, were collectively called junk DNA back in the day, which led Richard Dawkins to propose his selfish gene hypothesis, the idea that DNA appears to act for the purposes of its own preservation and propagation, rather than as merely a means to an end for producing the phenotype of a body. There is some evidence that segments of non-coding DNA actually help some genes to get expressed more easily by exposing them more openly on the chromosome, so it can have a functional purpose in eukaryotes. It's also possible that for prokaryotes, the economy of fast replication caused the increase in genome size to be selecting against the accumulation of unused DNA, and that might account for the lack of introns and other non-coding DNA in bacteria. An additional paradox is the relationship between the number of protein-coding genes and organismic complexity. There is a relationship between genome size and cell size. Species with bigger genomes have bigger cells, but the so-called G paradox describes the mismatch between the number of protein-coding genes and organismic complexity. The model organism Caenorhabditis elegans, which is a soil-living roundworm, has about the same number of genes as humans do, despite their having a mere 959 somatic cells. Think about that for a second. This is a metazoan, a multicellular eukaryote, with a fixed number of cells incapable of growth past that. There are plenty of animals that have determinate growth, like mammals, where we don't get larger past a certain point in life, but an actual fixed number of somatic cells whose developmental lineages have all been traced by developmental biologists, I think that's an amazing natural phenomenon and a real achievement in cellular biology to understand it. What explains this paradox is likely that it's not merely the number of genes, but also the network of regulatory elements like transcription factors that are important. When I was in grad school in the early 21st century, I always had the impression of this self-congratulatory nature of those genome sequencing pronouncements, like they were getting to the machine code of living things and the rest of us were just stamp collecting. It seems like in the end, once again, it's organisms that are the important thing, not merely genes, if we care about evolution. So, going forward, organismic complexity will probably be better understood by being able to census the entire proteome rather than the genome alone. So, everybody back in the lab.